Right now, inside your body, there's a whole universe of activity. Billions, and I mean billions of times every single day, your cells are dividing. They're copying themselves to keep you alive, to keep you healthy. It's this incredibly precise, microscopic ballet. But what happens when one of those dancers misses a step?
See, that constant process of cell division, it's not always 100% perfect. Little errors can sneak into the genetic blueprint, creating what you could call rogue cells. Now, most of the time, your body's got it covered. It cleans up these mistakes. But every once in a while, one of these errors can be the tiny spark that starts a much, much bigger fire.
And one of the most dangerous kinds of errors is something called a chromosomal abnormality. Just think of it like a huge, glaring typo in the cell's instruction manual. We're not talking about small mistakes here. These are major rewrites to our DNA. And they are a key driver behind some of the most aggressive cancers, the kind that spread, resist treatment, and come back.
But here's the massive challenge for scientists. These initial dangerous mistakes are incredibly rare, and they're totally spontaneous. So trying to find that one single cell that's making a mistake among, say, tens of thousands of perfectly healthy ones, it's like trying to find one specific typo in an entire library full of books. So how on earth do you study something you can barely even find?
That right there is the central puzzle that's been stumping scientists for ages. If you really want to understand where cancer begins, you have to be able to catch these rogue cells right in the act. And to do that, well, you need a completely new way of looking at things.
So, to take on this incredible challenge, researchers over at the European Molecular Biology Laboratory developed a totally groundbreaking new tool. They call it the MAGIC platform. And let me tell you, it is a brand new way to go hunting for these rogue cells.
MAGIC is actually an acronym, and it perfectly spells out what this thing does. It's a convergence, a fusion of high-powered microscopy, that's the imaging part, with incredibly sophisticated AI, which is the machine learning, and super detailed genetic analysis, that's the genomics. It brings all these amazing technologies together into one autonomous system.
Okay, probably the best way to get your head around how this all works is to picture MAGIC as a highly specialized microscopic SWAT team. It has a very clear mission: find, tag, and extract specific rogue cells for interrogation, all without bothering any of the healthy cells around them.
All right, so let's dive into the mission specifics. How does this microscopic SWAT team actually get the job done? It's a pretty brilliant four-step process.
This here gives us the bird's eye view of the whole operation. You see, it starts with just a sea of ordinary, unlabeled cells. Then, through this whole automated process, it ends with the detailed genetic blueprints of just a few very specific, very suspicious cells. It's a journey from the crowd to the individual.
Let's break that down using our SWA team analogy. Okay, step 1, surveillance. A super high-speed automated microscope is constantly scanning tens of thousands of live cells. Then, step 2, target ID. As all those images are flooding in, a machine learning algorithm, our AI analyst, is scanning them in real time, looking for any visual sign of trouble. Step 3 is tagging. The second the AI spots a target, it commands the microscope to aim a tiny laser and permanently tag that one specific cell with a special dye. And finally, step 4, extraction. All the cells that got tagged are then separated from the rest, ready for their genetic interrogation.
So what is this AI analyst actually looking for? What's the clue? Well, its prime target is something called a micronucleus. You can see them here. They're these small, separate little blobs of DNA that are sitting outside the main nucleus. And they are a classic telltale sign that a cell's genetic material is unstable, a warning flare that major chromosomal errors are probably on the way.
And check this out. This slide shows that tagging step in action. On the left, you see the before shot. The cell's nucleus is glowing green, pretty normal. But then, after the AI flags it as suspicious, a laser zaps it with this special dye, and poof! On the right, that exact same cell's nucleus is now glowing magenta. It's been marked. The SWAT team has tagged its target.
So, once you've found these incredibly rare rogue cells and pulled them out of the crowd, what do you do with them? Well, this is where the real payoff comes in. Scientists can now perform what's called single-cell sequencing. They can literally read the entire genetic code of each individual cell to see precisely what typos and errors have happened. And the first results from this were fascinating. For the first time ever, researchers could actually figure out a baseline rate for how often these mistakes happen. And what they found was kind of staggering. In a normal population of cells, a little more than 10% of all divisions result in some kind of spontaneous chromosomal abnormality. That's way higher than anyone thought, and it suggests our bodies are constantly, quietly dealing with these potential threats. But here's where it gets really crucial. What happens when you introduce a known risk factor? So the researchers ran the MAGIC platform on cells where they had disabled a really famous tumor suppressor gene, one called TP53. And get this, the rate of chromosomal errors almost doubled. This gives us a direct, measurable link between a specific faulty gene and the rate of this dangerous genetic chaos. Now, hunting for these chromosomal errors is huge, don't get me wrong, but it's just the beginning. The true power of MAGIC is in how versatile it is, and this is where the story gets even bigger. Just like the researcher Jan Korbel explains here, the whole system is adaptable. Yeah, they trained the AI to spot micronuclei, but you could train it to hunt for almost any visual feature in a cell. Think about it. The first signs of aging, a viral infection, how a cell reacts to a new drug. You can train the SWAT team to look for any kind of target you want.
So the real takeaway is this. MAGIC isn't a one-trick pony. It's a launch pad. It gives scientists a brand new tool to explore the microscopic world, a way to directly connect what a cell looks like with what its genetic code actually says. This just blows the doors wide open for entirely new frontiers of research.
Which, it leaves us with this really tantalizing question. For decades, we've studied biology by looking at the average behavior of millions of cells all lumped together. But now, for the first time, we have the power to hunt down the outliers, the rare individuals, the very first ones to go wrong. What other secrets of disease and of life itself are just sitting there, waiting to be discovered, one rogue cell at a time?